It's a time. Okay, well, it's noon, so good noon, I guess. It is my great honor and pleasure to introduce Dr. Kiran Khush, whom I've known for many years now. Dr. Khush is a professor of medicine and transplant cardiologist at Stanford University, where she's been on faculty since 2008 after completing her training at UCSF. She also completed medical school at Harvard and undergrad at Stanford. She is the director of heart transplant research and the program director for the Advanced Heart Failure Fellowship. She's a highly regarded and successful clinical and translational investigator in our field. She is NIH funded for heart transplant research, which is somewhat rare, and runs a wide-ranging research program with projects related to evidence-based evaluation and selection of donors for transplant, the development and testing of novel genomic biomarkers, novel therapies for prevention of vasculopathy, and development of assays to assess the immune environment. In addition, Dr. Khush serves as an associate editor for the American Journal of Transplantation. She serves on the SRTR National Transplant Program Review Committee and is the incoming program chair for the ISHLT 2025 Annual Scientific Sessions. She's a mother of three, and I recently learned that she's also an avid skier. The heart transplant field is akin to a large family, and in this family, Kiran is one of my favorite siblings. So please extend a very warm welcome to Dr. Khush.
Thank you so much, Monica. That was a very kind introduction, and it's really an honor and a pleasure for me to be here today. Talking about one line of research that I never would have thought that I would have gone into. So I think this is a great lesson for the fellows and junior faculty in this group to keep an open and curious mind, because you never know where your career may take you. So I'll be talking about clinical applications of cell-free DNA testing after heart transplantation. Let me try this one. Perfect. Okay. So these are my conflicts of interest. I do serve as a speaker and advisor for CareDx, which is a medical diagnostics company. I have an NIH grant related to using cell-free DNA for early cancer detection and a patent related to cell-free nucleic acids for analysis of the human microbiome. So general outline of my talk. I'll be talking a little bit to frame the discussion in the beginning about some background and historical perspective on cell-free DNA testing. The first part of my talk will be about cell-free DNA for surveillance of acute rejection after solid organ transplant. Then I'll be talking about cell-free DNA to monitor the microbiome, to screen for infectious complications, and cell-free DNA for early diagnosis and detection of malignancies after transplantation. And finally end with a few words about cell-free DNA as an actual mediator of the alloimmune response. So to start out with, cell-free DNA is a very powerful biomarker. So for those of you who may not be familiar with this field, cell-free DNA is released from any nucleated cell in the body via normal cell turnover or apoptosis, or through tissue damage such as necrosis. It can be found in any body fluid, serum, plasma, CSF, urine, and so on. And it's derived from genomic or mitochondrial DNA. And the fragment lengths vary quite a bit, from very short to quite long fragments. And that may give you a clue as to the etiology of the cell-free DNA. And finally, it's a very dynamic biomarker because it's cleared from the blood within about 15 to 30 minutes. So it really reflects the processes going on in the tissues in the body at that particular snapshot in time. So a little bit of a historical perspective. Cell-free DNA was first described by a group of French scientists in the late 1940s. And in 1966, DNA was identified in the plasma of patients with lupus, and it was found to be quite elevated in the setting of lupus flares. And now we know that cell-free DNA is released from the tissues in the setting of tissue injury or damage. In the 1970s, cancer patients were found to have very high levels of circulating cell-free DNA. And we now know that we can actually detect and quantify circulating tumor DNA in our patient's blood. And finally, in 1997, the field really took a huge step forward with the identification of fetal DNA in the maternal circulation during pregnancy. And this was actually a breakthrough made by Dennis Loh, who's a professor at the Chinese University in Hong Kong. And remember Dennis Loh, because I'll come back to him at the end of my talk. But where I started to get involved was that I met Steve Quake, who's a professor of bioengineering at Stanford. Steve is a real entrepreneur. He takes sequencing methodologies and he devises or he looks into medical applications of his new sequencing techniques. Steve started a company that many of you may have heard of called Fluidigm. So Steve was giving a talk on his development of one of the first prenatal cell-free DNA assays. And in hearing about his work, we started wondering, could the same situation be true in transplantation? So just as you can detect DNA from the fetus, or more accurately from the placenta, in the maternal circulation during pregnancy, can you actually detect DNA from the donor organ in the recipient's blood after solid organ transplantation? And could this be a marker of graft injury or potentially graft rejection? So we went to Steve with this question and he said, well, that's a really fascinating question. I have no idea, but let's give it a try. And so we pulled some banked plasma samples we had from previous heart transplant research studies that we've been involved with from patients with and without acute rejection and showed that we actually were able to detect and quantify DNA from the donor in our transplant patient's blood. And it seemed to go up in the setting of acute rejection. So this set the stage for a prospective study that we did looking at cell-free DNA as a non-invasive marker of acute rejection after solid organ transplant. My talk today focuses on heart transplantation, but this is relevant to any solid organ transplant. And similar work has been done in lung and liver and kidney transplantation as well. It's a universal biomarker. So just to introduce the concept, you can think of a solid organ transplant as a genome transplant. So when you're transplanting an organ from a donor into the recipient, cells from that organ are going to turn over, whether it's through apoptosis or necrosis, and release a small amount of DNA from the donor into the recipient's blood. And if we know the sequence differences of the donor and the recipient, we can actually sequence the cell-free DNA floating in the plasma and figure out which cell-free DNA fragments are coming from the donor and which are coming from the recipient. And that's really kind of the background for the work that we did. So our hypothesis was that in the healthy state of immune quiescence, a very small amount of the circulating cell-free DNA in a transplant recipient is going to come from the donor organ. But in the setting of acute rejection, where you have damage to the graft and graft injury and cell death, a larger amount of DNA circulating in the blood is going to come from the transplanted organ. And if we can actually then identify which are the donor DNA fragments, we can quantify them over time and see if they go up in the setting of acute rejection. And so we received a 3-year grant from the NIH to do a prospective study where we enrolled 160 adult and pediatric heart transplant patients, and we collected blood from the donor and the recipient at the time of transplantation. We then sequenced the donor and recipient DNA to identify SNPs where they had sequence differences, and that allowed us to determine which cell-free DNA fragments were coming from the donor and which came from the recipient after transplant. We then collected serial plasma samples from the recipient after transplant and isolated and quantified the DNA coming from the donor organ and correlated it with clinical events such as development of acute rejection. So what we saw was that in the first few days after heart transplant, there's very high levels of DNA coming from the donor organ in the recipient's blood. And that's not surprising because the graft undergoes a fair amount of injury from procurement and cold storage and ischemia reperfusion injury. So right after transplant, a lot of DNA from that donor organ is being released into the recipient's blood. But because it's cleared so rapidly, it falls to a low baseline level within about several days to a week after transplant. And it remains at these very low levels in the absence of acute rejection. But what we found was that in the setting of acute rejection, and this is a patient with a moderate and a severe cellular rejection, the amount of DNA coming from the donor organ increases significantly in the recipient's blood. And this is also true of antibody-mediated rejection because both processes cause cell injury and cell death. And so what we found was that in aggregate, when we looked at all of our patients, the amount of DNA coming from the donor organ increased significantly in the setting of acute rejection. We also saw that the donor-derived cell-free DNA levels started to go up in the recipient's blood weeks to months before rejection was diagnosed on biopsy. And we now know that molecular markers are actually a more sensitive marker of graft injury and acute rejection than biopsy, because by the time we're seeing cell death on biopsy, it's relatively late in the process of alloimmune activation and graft injury. And so actually these cell-free DNA assays are more sensitive than biopsy for picking up acute rejection. And I think this offers us a chance to be more proactive clinically in the treatment of our patients, where if we see the cell-free DNA levels start to rise, we can actually augment immunosuppression or start treatment earlier to prevent a severe rejection that may cause irreversible graft damage. So we've all seen cases where technology developed in one center could not be replicated or was not translatable to another site. So around this time, my mentor, Hannah Valentine, who'd worked with me on development of this assay at Stanford, moved to the NIH and started a lab over there. And so Hannah, along with Sean Agborino, who's a lung transplant physician at the NIH, actually tried to replicate these studies. So we sent them plasma samples from our patients with and without acute rejection. They developed their own cell-free DNA pipeline and reran these samples and got identical results. And so this shows that this is an assay that's very simple in concept and easy to reproduce and can be performed really at any site that has sequencing and informatics capabilities. And so that was the first barrier that was overcome. Hannah and Sean subsequently launched a multi-center study called the GRAFT Consortium, where they collected serial plasma samples from heart transplant patients at 5 different centers, ran this donor-derived cell-free DNA assay, and had very similar results, that the donor-derived cell-free DNA levels go up significantly in the setting of acute rejection and also correlate with the severity of graft dysfunction. And the area under the curve was excellent, about 0.9 for rejection surveillance. And so this was really how this non-invasive assay for rejection surveillance was developed and launched. But the next step, and sorry, just getting back for a second, Hannah and Sean actually looked at fragment lengths of cell-free DNA and showed that the fragment sizes were shorter in the setting of antibody-mediated rejection compared to cellular rejection. So for the first time, we saw the possibilities of starting to discriminate different types of rejection based on non-invasive monitoring. To date, what we've done is we've used non-invasive monitoring to screen for rejection. And if these tests were abnormal, we go on to do a biopsy to confirm that rejection was present and determine the type of rejection, which dictates our treatment algorithms. But now we may actually be getting into an era where we can determine the type of acute rejection just based on our molecular assays and forego biopsies completely.
Yeah, do what?
So there's different cell types that release cell-free DNA. In acute cellular rejection, it's being released from myocytes. Antibody-mediated rejection, probably more endothelial cells that may have to do with the underlying pathophysiology. They also showed that cell-free DNA levels started to go up earlier and were much higher in the setting of antibody-mediated rejection. So the technology has changed a lot over the past 10 to 15 years. The approach we used was called shotgun sequencing, where you sequence all of the cell-free DNA in the patient's blood. Over time, other centers started to do more targeted assays, looking at particular SNPs instead of whole genome sequencing. Informatics algorithms started to be developed to accelerate the analysis of this data. And so this field has really moved forward quite quickly until now we have these automated systems where basically you're allowed to increase the throughput of these assays dramatically through automated nucleic acid isolation, library prep, sequencing, and informatics. The stage was set for development of a clinically available donor-derived cell-free DNA assay. And so the license for our assay was actually acquired by a company called CareDx, and we started working with them on development of a clinical donor-derived cell-free DNA assay. So the first assay that was developed, there's now 2 that are clinically available in the US, was called AlloSure, made by CareDx. And it's a much more simple and streamlined platform. So instead of sequencing all of the donor and recipient DNA and looking for their sequence differences, what this assay does is has a panel of 405 highly polymorphic SNPs. So these are sites in the genome that are reliably different between 2 different individuals. If you just sequence at these 405 SNPs, you can differentiate DNA from 2 different individuals, even related individuals, such as in the setting of living-related kidney transplantation. So they sequence these 405 SNPs, it goes through an informatics pipeline, and that calculates which, it determines which DNA fragments are coming from the donor, which are coming from the recipient, and it calculates the donor fraction, and that's the information that's transmitted back to the transplant center within about 48 to 72 hours. So relatively rapid turnaround, which is important for a clinically relevant assay. So I was involved in the DOR registry, which is a large prospective study that attempted to determine the clinical utility of this donor-derived cell-free DNA assay. So the DOR study was performed at 26 heart transplant centers in the US, 740 patients, where thousands of blood samples were collected for cell-free DNA analysis, and many of these assays were paired with biopsies. So 840 samples were paired with biopsies, and out of these, there were 18 AMR, or antibody-mediated rejection, and 17 cellular rejection events. And what we found from this large registry was that in the absence of rejection, donor-derived cell-free DNA levels stay very low and stable, with a median level of about 0.06% for at least up to 2 years after transplant. But in the setting of acute rejection, we saw significant increases in the donor-derived cell-free DNA levels using this clinical assay. And intuitively, the levels go up as the rejection becomes more severe. So as the rejection is more severe, you have more graft damage, you have more cell death, more donor-derived cell-free DNA is released into the blood, and the levels go up in the blood. And this is true for both cellular and antibody-mediated rejection. We also looked at patients who were treated for graft dysfunction, who had negative biopsies, and we found that their cell-free DNA levels were significantly elevated. And so this speaks to the fact that the cell-free DNA assay may actually be more sensitive than the biopsy for detection of acute rejection. And we now know that biopsies have many limitations. There's sampling error, where you may be picking a piece of myocardium that doesn't have rejection when it's in fact present. There's a lot of variability between pathologists in terms of biopsy grading. So the biopsy really has turned out to be a fairly poor gold standard, and maybe the molecular assays will actually be better, more sensitive at picking up graft injury and graft damage than the biopsy. So overall, this is the AlloSure test performance. I think the most important characteristic for surveillance assay is the negative predictive value. So if the assay is below threshold, how confident can you be that acute rejection is not present? And in this case, the negative predictive value is 97%. So if the AlloSure assay is less than the threshold of 0.2%, we're 97% sure that the patient does not have rejection and does not need to have a biopsy. And so this has really revolutionized the field of heart transplant surveillance. It used to be that most centers, ours included, would do 12 to 18 routine heart biopsies within the first year or 2 after transplant. And obviously, patients don't like it. We don't like doing all these biopsies. It can cause complications from this invasive procedure. And with the advent of these non-invasive molecular assays, we've been able to scale back significantly to the point where now we only do 2 routine biopsies at Stanford, and then all of our patients get switched over to non-invasive surveillance. And these assays have been adopted by over 90% of all heart transplant centers in the US. It's really changed the way we care for our patients. So common clinical questions that arise with the use of these assays. One is, a lot of patients say, you know, my AlloSure assay is positive, but the biopsy is negative. What should I do? Well, there's always a chance that it could be a false positive, but what I tell people to keep in mind is the fact that these assays start to go up weeks to months before the biopsy turns positive. So if you see an upward trend in your AlloSure results, this may be a patient with impending rejection. They may already have some degree of immune activation and graft injury. Now, how about the patients who really don't have rejection and have elevated AlloSures? So we've seen several different clinical scenarios. A lot of these are the patients with early post-transplant restrictive physiology, elevated filling pressures. You can get high donor-derived cell-free DNAs, probably from some subendocardial ischemia in the time of volume overload. We've also seen that these AlloSure levels start to go up in the setting of development of de novo donor-specific antibodies. And this is important because for many of us, for many years, we'd be monitoring DSAs in our patients and they would come up positive and we didn't know what to do about it. Should we treat these DSAs or not? Should we just watch them? And now we know that with development of pathologic DSAs, the cell-free DNA levels from the donors start to go up in the recipient's blood, and this can actually help us guide our treatments. And so patients who have DSAs with rising cell-free DNA levels may actually warrant therapy to prevent further DSA production. How about cardiac allograft vasculopathy? So the most common long-term complication after transplantation, this can cause graft ischemia. The data on CAV and cell-free DNA is still mixed. There was a small study from the University of Chicago showing that patients with elevated cell-free DNA levels tended to have more allograft vasculopathy, but then there was a study from Spain using a different assay, but the same concept showing that there was no difference in donor-derived cell-free DNA levels with development of CAV. We recently completed a study at Stanford and Intermountain Medical Center where we again saw no elevation in donor-derived cell-free DNA levels with development of cardiac allograft vasculopathy. And I think that's because most of these patients still were asymptomatic with normal graft function. I think it'd be interesting to actually study patients with graft dysfunction in the setting of severe CAV to see if they start to leak cell-free DNA into their blood. How about race? This is a topic I've become very interested in recently. Getting back to the graft consortium run by Sean Agborino at the NIH, what they found was that patients of Black race have higher donor-derived cell-free DNA levels within the first week after transplantation compared to white patients. And this may be because Black patients may have more early graft injury. They tend to have more race and HLA mismatch between the donor and recipient. But the question I ask is, does this set them up for subsequent graft damage further down the road? He also showed that Black patients tend to have higher donor-derived cell-free DNA levels 6 to 12 months after transplant compared to white patients. And I'm going to get back to this later in my talk today, because I think cell-free DNA may actually be mediating further graft injury and further graft damage in these patients and may be contributing to the worst transplant outcomes that we see in our Black patients. How about sex? This is hot off the press. This paper was just accepted to Journal of Heart and Lung Transplant 2 days ago. We should always look at sex differences in any assay that we develop. We did not find any sex differences in donor-derived cell-free DNA levels, either in the presence or absence of acute rejection, and the assay performance for surveillance was similar in men and women. So where is this field going in terms of acute rejection? One patient population that's excluded from using cell-free DNA assays is our multi-organ transplant patients. And the reason is we can detect cell-free DNA in the blood, but we can't tell you which organ it's coming from. And that's important because different organs have different thresholds. So for example, the threshold we commonly use after heart transplant is 0.2% for the cell-free DNA. In lungs, it's 0.5 for single lungs, 1% for double lungs. In kidney, it's about 0.5%. The liver is a huge organ that's highly vascularized and releases a lot of cell-free DNA from the donor organ into the blood, just in the setting of quiescence. So a cell-free DNA level of 5% can be normal for a healthy liver transplant. So if you have a heart liver transplant, you get a cell-free DNA level and the level comes back at 3%. You have no idea how to interpret that. Is that a heart transplant rejection or is it a normal liver? So we can't use these assays for multi-organ transplantation. So what some groups are looking at now is differentially methylated regions. So different cell types have differentially methylated regions on their cell-free DNA. And can we leverage this to determine the tissue specificity of the cell-free DNA? So which organ it's coming from. And I think we're getting closer and closer to being able to do that. And hopefully then we'll be able to distinguish the cell type of origin, not only to be used for multi-organ transplant, but again, to help us distinguish rejection subtypes, such as cellular versus antibody-mediated rejection. So to kind of switch focus a little bit, I talked a lot about acute rejection, but there's so many other clinical applications of cell-free DNA. And what I'd like to spend a few minutes talking about is sequencing of the microbiome or predominantly the virome, which is the work that we've been involved in. So as any of you who've taken care of transplant patients know, we walk a really fine line between over and under immunosuppression. You over immunosuppress your patients, they end up with all sorts of infections. You under immunosuppress them and they develop acute rejection. So we're trying to always target this very narrow therapeutic window. And I think that, one of the holy grails in transplantation is to develop an assay that allows us to determine how immunosuppressed our patients are, which varies a lot from one patient to another on the same therapies. So when we were doing this initial cell-free DNA work, we found that we were picking up about 2% of the reads we were picking up were non-human. And we started aligning these sequences with reference databases, and we found that these cell-free DNA fragments were coming from different microbial pathogens, bacteria, viruses, and fungi. So what we did was we took a deep delve into the viral sequences we're finding in our patients' blood. And we found all sorts of different viral sequences, but the predominant type of virus in our patients' blood came from a family called anelloviruses or torquetinoviruses. And I had never heard of this type of virus before, so I had some learning to do. And what I learned was that these are viruses that are found in the blood of about 50 to 90% of warm-blooded animals, 90% of humans in some region of the world. They have no known pathogenic role, so they're not known to cause disease, but these viruses kind of lay dormant and then replicate in the setting of immunosuppression. So for example, patients with HIV, as they progress towards AIDS, the relative amount of anellovirus in their blood goes up dramatically. So we started interrogating the virome in our heart, and we included some lung transplant patients as well. And what we saw was that as our patients became more immunosuppressed, the amount of anellovirus in their blood went up dramatically. So as you can see here, if you kind of go down the y-axis, as our patients' tacrolimus levels went up as they got more immunosuppressed, the relative amount of anellovirus, which is shown in orange, went up. We also saw responses to antiviral therapy. So we use valganciclovir for prophylaxis against herpes viruses like CMV. And we found that as we increased the amount of valganciclovir that we gave in our patient, the relative amount of herpes virus DNA in the blood, shown in the dark blue, went down. So you can actually see changes and fluctuations in the virome in our patient's blood in response to the treatment that we're giving them. And so we then looked longitudinally. So this is kind of a group of heart transplant patients where we interrogated their virome on the day of transplant. And they had all sorts of different viral sequences in their blood, herpes viruses, adenoviruses, cautoviruses. But over the first 6 months after transplant, as they became very highly immunosuppressed, the relative amount of anellovirus in their blood increased dramatically. And then over the next 6 months, as we weaned their immunosuppression, the amount of anellovirus went down. So we started to think about, could the amount of anellovirus in our patient's blood be used as a marker of how immunosuppressed our patients are? And it turns out that it can. So when we looked at patients with and without acute rejection, we found higher anellovirus levels in patients who did not reject, who are more immunosuppressed, and lower anellovirus levels in our patients with acute rejection who are less immunosuppressed. So maybe the amount of vanillovirus in our patient's blood could actually be used as a marker of overall immunosuppression, and finally kind of get us towards that holy grail of figuring out how immunosuppressed our patients are. Now, it turns out it's a little bit more complicated because there's 96 genotypes of vanilloviruses, and so we don't quite know which genotype to amplify, and there's cross-reactivity and sequencing with other viruses. So we're still a little bit a ways from developing a clinical vanillovirus assay, but there's multiple groups working on this problem. We also found, excuse me, when we were sequencing our patient's virome, that we were picking up sequences from CMV and looking more broadly from Pseudomonas and Staph. And so we started to see, do these actually correlate with our clinical lab results? So if we had ordered a CMV PCR, do the CMV levels that we pick up through our shotgun sequencing actually reflect the clinical lab values? And we found that there was a high degree of correlation between our results and the clinical lab results. So the next question we asked was, well, if we can just sequence, the virome, the microbiome in our patients, could we use this as a diagnostic tool to diagnose infections after transplant? Because as you all know, when a patient comes in, with a fever or signs of infection, what do? We order specific lab tests based on what we think they have. So we order a CMV PCR or a respiratory viral PCR or a COVID PCR. So your ability to diagnose an infection is directly related to how well you a priori decide which lab test to order. And often we're lucky and we diagnose the infection, but sometimes we never figure out what infection our patients have because we just didn't order that particular test. But now that we can actually just sequence all the microbes in our patient's blood, could we actually use that to cast a wide net and say what infectious agents are present in our patient's blood? So we used a cohort of lung transplant patients. The lung transplant patients come in all the times with infections. And we pulled some plasma from patients who came in with infections that were never diagnosed clinically. And we saw very high levels of certain viruses, adenoviruses, polyomaviruses, herpes viruses in their blood. And so this offers us a new way to diagnose infectious complications after transplant just by sequencing their microbiome. And so I had a couple of slides that I had to take out for CME reasons because it had company names on it, but this is a reminder to me. So one of the papers we published on sequencing the microbiome after lung transplant was led by a postdoc named Mickey Kurtz, who went on to start a company called Carius Diagnostics. And some of you may have heard of the Carius assay. It's basically a next generation sequencing assay you can use to diagnose infectious complications after transplantation. So if you have a patient who comes in with fever of unknown origin, you can send a Carius test and they just sequence the microbiome in the blood and send you back the results. And it can be helpful in these cases where you're having trouble coming up with a diagnosis. I think the problem is that you get a lot of noise. So you get tons of sequences, and then what's a bystander, what's actually a pathogen? It kind of takes a little bit of work to sort the wheat from the chaff. I did some work with CareDx, trying to also develop another next generation sequencing assay to diagnose infectious complications after transplant. That work is kind of on hold right now because the company we're partnering has been bought by Illumina. But we had a really interesting case I wanted to share with you where we had a heart transplant patient who came in with blurry vision 4 months after transplant, floaters. And after a whole series of tests, we did PCR of his vitreous fluid from his eye, and he was diagnosed with ocular toxoplasma chorioretinitis. And we had banked plasma samples of from this patient going back for months because we have a biobank at Stanford where we bank all of our patients' blood. So we went back through his plasma samples and we found toxoplasma DNA in his blood weeks before his clinical presentation. And actually, we were able to pick up toxoplasma DNA 2 weeks before the clinical lab toxo PCR assay turned positive. And so this is just one case, but it shows you the potential power of these sequencing-based approaches to diagnose infections, especially rare infections, which our transplant patients often have. And finally, one of the last clinical applications is cancer monitoring. So for those of you, who keep abreast of the oncology space, you'll know that the field of cell-free DNA has really taken great leaps forward in oncology. And this is really important for our transplant patients because, as you may know, transplant patients have a really high incidence of cancer development because the immune system doesn't only survey against, infections and rejection, but also against cancer development. So by 5 to 10 years post-transplant, cancer is a leading cause of death for our patients. And overall, transplant patients have a twofold higher incidence of cancer compared to the general population, and it's much higher for specific cancer types such as post-transplant lymphomas and skin cancers. And the problem with some of these cancers, like the PTLDs, the post-transplant lymphomas, is that they're often diagnosed very late because they present with very nonspecific symptoms, fatigue, night sweats, malaise. So by the time it's picked up, it's quite advanced. And so what we now know is that there's this concept of the liquid biopsy. So you can actually pick up DNA from tumors or circulating tumor DNA in patients' blood, and there's many different clinical applications. So it can be used for diagnostic purposes to diagnose cancers in a very early stage. You can also use this for prediction. For example, the genetic makeup of a cancer may help you determine prognosis or potential response to therapy. It may help you choose targeted therapies. And then you can also monitor for early relapse by monitoring for circulating tumor DNA. So many clinical applications of cell-free DNA within oncology. So I was fortunate to collaborate with an oncologist at Stanford named Ash Alizadeh, who does a lot of work on circulating tumor DNA for detection of lymphomas. So Ash has a strategy called CAP-seq, where he identifies mutations common to different lymphomas, and then also sequences tumors, identifies the cancer-specific mutations in tumors, and develops SNP panels of all these common mutations, which you can then use to screen circulating cell-free DNA to identify circulating tumor DNA and to hopefully identify cancers very early. And so we went back through some banked plasma samples we had. These are from a couple of lung transplant patients who went on to develop PTLD. And we found that we could identify lymphoma DNA in their blood several months or longer before the clinical diagnosis. So the figure on the left shows the clinical diagnosis in the dotted vertical line. And you can see that we were able to detect circulating tumor DNA, shown in blue, a month and a half before the clinical diagnosis. And then we were able to track response to therapy. So this patient was treated with rituximab for PTLD, and we saw a dramatic decrease in the circulating tumor DNA in response to rituximab therapy. On the right side, we have another lung transplant patient who was diagnosed with PTLD, where we could pick up circulating tumor DNA from the lymphoma over a year before the clinical diagnosis, and again showed a very positive response to rituximab therapy with disappearance of the circulating tumor DNA in the blood. This is another patient. This is a really interesting case. And so we had a patient who developed PTLD, and we monitored the circulating tumor DNA in response to rituximab therapy, showing that it went down significantly, but this patient did not have a complete response. So after therapy, the circulating tumor DNA levels went up dramatically again. Then the patient was treated with R-CHOP chemotherapy, and the circulating tumor DNA went away. And then finally, in this case, another patient with PTLD who had an excellent response to chemotherapy, but unfortunately, we scaled back a lot on his immunosuppression, and he then developed acute rejection, as shown by monitoring the donor-derived cell-free DNA levels in red. So this shows the power of this approach, where with a single blood test, you could screen for rejection, looking for donor-derived cell-free DNA. You can screen for malignancies, looking for circulating tumor DNA, and you could also screen for infections as well. And so really, I think there's so many fascinating clinical applications of this technology. So we now have a NIH grant to develop circulating tumor DNA assays for early detection of post-transplant malignancies. We're mainly focusing on PTLD, but also lung cancers and colorectal cancers, and also looking at the role of EBV, Epstein-Barr virus, in mediating the development of some of these malignancies. So we've partnered with many different transplant centers around the world because it's really hard to find banked blood samples from transplant patients before they develop the cancer. And we want to go back through these blood samples that precede the diagnosis to see if we can find cancer DNA in their blood before they present it clinically. But we have been able to collect about 70 PTLD cases and some lung and colorectal cancers. And we've been going back through their blood samples showing that we can pick up tumor DNA in their blood weeks to months, even a year before the clinical diagnosis. So our hope is one day we can have assays to screen for cancers in our very high-risk transplant patients and hopefully pick them up and treat them earlier and improve patients' outcome. We showed here that we start to see more and more EBV virus and more mutations in our patient's blood within 6 months prior to the diagnosis. And also we see EBV levels and adenovirus levels start to go up in our patient's blood. And this is really why heart and lung transplant patients get more malignancies compared to liver and kidneys, because they're more immunosuppressed, right? Because we don't HLA match hearts and lungs. And so they require more immunosuppression. As you can see here, they have much higher adenovirus levels compared to abdominal transplants. And this is why they're more immunosuppressed and develop more post-transplant malignancies. So finally, I'm going to end with a new area of research that I've become involved in, which I think is really fascinating, which is cell-free DNA, not only as a byproduct of graft injury, but as a mediator of allograft injury. And this is a relatively new concept. And I know this is kind of a busy slide, but what I want to highlight here is the fact that when a heart is procured and transported and then transplanted, it undergoes a fair amount of graft injury. And so we know that after transplantation, it releases a lot of cell-free DNA into the blood. And there's been a lot of data recently that cell-free DNA itself may be what's called a DAMP, or a damage-associated molecular pattern. So these are molecules that activate the innate immune response by binding to receptors such as toll-like receptors and pattern recognition receptors on innate immune cells, dendritic cells, NK cells, and so forth. And then this really causes a whole inflammatory signaling cascade with release of inflammatory cytokines, activation of the adaptive immune response, and so forth. So could cell-free DNA not only be a byproduct of graft injury, but could it also perpetuate the immune response and cause more graft injury down the road? And so, one question that comes up is, could this be why Black patients have worse outcomes after transplant? Because they have higher cell-free DNA levels in their blood in that first week after transplant. And is that setting up a vicious cycle of immune activation and cell damage? And so there's now an increasing body of work, mainly pioneered again by Sean Agbar-Eno, showing that cell-free DNA arising from these innate immune cells can cause organ injury. And Sean did a really elegant series of experiments where he had cell cultures of neutrophils, vascular endothelial cells, and other cell types. And he then treated these cells with just pure cell-free DNA isolated from patients during acute rejection and no rejection, and showed that cell-free DNA in and of itself coming from patients with acute rejection caused cell lysis. And that's shown here. These pink cells are pink because they're releasing reactive oxygen species. So this is direct evidence that cell-free DNA in and of itself can cause cell death and graft injury. So this is important because it potentially affords us new ways to immunosuppress our patients. So we know that there's these DAMP pathways where cell-free DNA and other DAMPs activate the alloimmune response through different signal transduction cascades, but there are also molecules that block these signal transduction pathways. So there's what's called DAMP pathway inhibitors. So could we actually use DAMP pathway inhibitors to immunosuppress our patients, especially our high-risk patients who develop a lot of rejection? Could this be a novel therapeutic option for our patients? Because as we know, we've been using the same immunosuppressants now for the past 20 years or longer, and could DAMP pathway inhibitors be a novel way to immunosuppress our patients and prevent graft damage and graft injury? So this is a very rapidly growing field. I just did a simple PubMed search looking at cell-free DNA and transplant. And we started that first proof of concept paper on the ability to detect and quantify donor-derived cell-free DNA in 2010. And excuse me, now we're in this exponential growth phase where there's more and more studies related to cell-free DNA and transplantation being published every year, close to 100 papers last year. And so I think this is really exciting because, excuse me, there's so many different groups working in the cell-free DNA space, lots of new ideas, new development, new clinical applications. And so it's really been a fun area for me to be involved in. And again, not where I expected my career to go, but it's been a great learning experience for me. So back to Dennis Lowe. So I think a recognition of the importance of cell-free DNA as a diagnostic in medicine came about in 2022 when Dennis Lowe was awarded the Lasker Prize, which is often considered to be a prequel to the Nobel Prize for his work in cell-free DNA testing. So in conclusion, I hope I've shown you that donor-derived cell-free DNA enables the non-invasive monitoring for acute rejection after solid organ transplant. It's certainly changed the way many of us care for our patients. And hopefully one day we'll be able to distinguish acute rejection subtypes, tissue of origin. And I really think we're on the road to completely replacing endomyocardial biopsies with molecular assays. The anellovirus load could eventually be used to measure the overall level of immunosuppression in our patients, which may help us personalize, better titrate immunosuppression to our transplant patients. And also, non-human DNA can be sequenced to screen for and diagnose infectious complications after transplantation. Circulating tumor DNA can be used for early detection and monitoring of post-transplant malignancies. And finally, cell-free DNA in and of itself may be a mediator of allograft injury by activation of DAMP pathways and may lead to new therapies to prevent acute rejection after solid organ transplantation. So with that, I'd like to acknowledge my many collaborators and folks I've worked with over the years who've contributed to this work. I've learned so much from them, and I'd like to thank all of you for your invitation and for your attention today. Thank you.
Awesome. Amazing presentation, Dr. Khush. An absolute tour de force of a new technology that has come so far in the last 10 years. We have some time now for questions. If anyone from the audience, Keith.
I'll stop. So first of all, that was. Oh, there you go. Thank you. That was spectacular. I'm literally totally biased, but by my interest in all this, but it was spectacular. And the fact that you've taken this technology and identified the possibilities of really, I mean, just fully interrogating the post-transplant experience, at least from a biological standpoint, is remarkable. That said, the technology is further along with respect to rejection than it is with respect to infection and malignancies. And as you very well showed, we're always trying to find that right balance, right, between over and under immunosuppression. But in fact, we've tended toward some over immunosuppression because we have more people dying of infection and malignancy than we have dying of rejection. So this is a loaded question, but you alluded in the very beginning that something we all know that cell-free DNA will go up. It's a harbinger of what's to come, right? You can see it long before someone actually rejects. And I suspect there will be a tendency for people to augment immunosuppression when we see cell-free DNA going up. But yet this is all in the context of knowing that we have more people dying of infection and malignancy than we do of rejection. So are there any prospective plans to try to provide guidance with those considerations in mind?
Right. So I see where you're coming from. I think the flip side is that because it's just a blood test, we can do it a lot more frequently than we could do a biopsy. And hopefully in the stable state, we can wean immunosuppression based on the results of these tests. And that's what I'm actually hoping will happen, that say you did a cell-free DNA assay every 2 weeks or so and it was stable. Can we actually be more aggressive in weaning immunosuppression? Because then you can get a follow-up level in a week or two. If it starts to go up, then maybe you go back up. So could it actually allow us to personalize our immunosuppressive therapy by more finely titrating or modifying our immunosuppression based on what someone's cell-free DNA levels are? And so actually, along with, Jeff Tudeberg and some of my other colleagues were trying to design a trial where we titrate immunosuppression based on cell-free DNA levels and actually get away from biopsy completely. So if the cell-free DNA levels start to go up, if we give an empiric short course of steroids and they come back down, can we then monitor that and then continue to wean immunosuppression with the hope of eventually getting patients on less immunosuppression? Because, for example, right now, as you know, when we back off on immunosuppression, say a patient has a bad infection or a cancer, we do so somewhat gingerly or reluctantly because we're worried that they may reject. But now we could just send a cell-free DNA assay a week or two later and see if it's going up or not. And so my hope is ultimately it will allow us to use less immunosuppression, especially once you get past that early 6-month period.
Off more than already do.
Right.
Stabilize.
Exactly.
Thanks.
Please.
Hi, thank you for that outstanding talk and sharing all your work. You alluded to this a little bit during your talk, but what do you do for the patient that does have a higher, donor fraction than expected, very much in the quote unquote rejection range, but hemodynamics are perfect and graft function is great and patient's doing fine. Do you put that patient kind of on steroids just because like the cell-free DNA is at 1% where otherwise biopsy is negative, everything else is perfect?
Yeah, so I have had a few patients who I've treated empirically. So, in a couple of cases, I've had patients who did not want to do a biopsy at all. And some of them were really sick in the ICU. We couldn't do a biopsy. We had some patients who were just terrified of doing biopsies, who refused to do a biopsy. And I have treated patients empirically and given them a pulse of steroids and then followed the cell-free DNA levels because they start to go down pretty quickly after rejection treatment. So I have used it that way. But we've all had patients where we've had a high cell-free DNA level and we're all scratching our heads like, why is it so high? We did a biopsy, it's normal, they're asymptomatic, they have normal graft function. So I think we're still kind of learning more and more about what this means. I do check DSAs, because if their DSAs start to go up, and sometimes I'll even send for non-HLA antibodies, because we know that as they start to develop these pathologic assays, their cell-free DNA levels can start to go up. But certainly I've had patients who have just elected to watch, and maybe I'll check a follow-up level a week or two later. And if it's coming down, I feel reassured. And if it keeps going up, then I may just treat empirically. So I think we're still learning about some of those cases. I also am not entirely sure if we have a patient with renal failure on dialysis, how the timing of dialysis impacts their cell-free DNA levels. So that's a study that we're still hoping to do.
Any questions for Dr. Colleen?
Thank you for the fantastic talk. I really enjoyed it. That you addressed what this implications this might have on your use of endomyocardial biopsy. How about the use of AlloMap? Does it impact the value of AlloMap on clinical decision making?
Yeah, so that's a great question. So, AlloMap is the peripheral gene expression profiling test that's also available for heart transplant monitoring. And right now, the way CareDx is kind of offering these assays is that you order it together. You get the gene expression test and the cell-free DNA assay at the same time. It's called HeartCare. And a lot of people wonder, do I even need AlloMap anymore? And what we know is that there are a lot more false positives with AlloMap compared to the cell-free DNA. So the AlloMap test can be elevated in the setting of infectious complications. It's a gene expression test. So immune-mediated genes get activated in the setting of CMV infection, for example. So if anyone has CMV viremia, their AlloMap may go up. But there's many other reasons why the AlloMap test may be positive. So I tend to rely more on the cell-free DNA result than the gene expression result when I'm interpreting the results of these tests. but actually, we just submitted a manuscript for publication looking at combined testing compared to either one in and of itself. And the positive likelihood ratio for both tests being positive is a lot higher than either test in and of itself. And so if both tests are positive, you're much more likely to have a positive biopsy compared to if just an AlloMap or just an AlloSure is positive. And I think in general, where the field is going is different molecular assays are being developed to look at different facets of the alloimmune response and different complications. So we're going to have assays one day to look for CAV. We have assays that look for graft dysfunction. So how do we best combine these molecular assays to get the most information? I don't think it's just going to be one assay or the other, but rather how can we use these assays in combination, because can they be complementary? So the gene expression test looks at immune activation. The donor-derived cell-free DNA test looks at graft damage. So in combination, could they be more informative than one assay on its own? And I think the answer is probably yes.
So Keith and I were talking before you brought up the Curious test, because that was the first thing, or one of the things that came to mind, and how sensitive it is and how it's helped us in these clinical conundrums where we're not sure what's going on with the patient. Is there any additional testing that you're currently doing if you have someone who has an isolated plasma cell-free DNA, all their testing's been negative, you worked them up for CAV, you've done everything else that you normally would do? Is there additional testing that you're currently doing?
I'd say the one thing that I sometimes consider doing is sending out for non-HLA antibodies. Just because we're learning more and more about the potential pathogenic role of different non-HLA antibodies. But it's hard because, at least for us, it's a send out lab. It takes like a month to come back. So it's not very clinically useful in real time, but it is another piece of data that I've started incorporating kind of when you have these perplexing cases where you're not quite sure what to do with it.
That was a phenomenal presentation. It seems like this technology, when and if it continues to advance, may become the gold standard to assess rejection. Can you walk with me a little bit about what do you think should happen research-wise, the company investment-wise, healthcare-wise, for that to become the gold standard test? Kind of what steps needs to occur?
Yeah, great question. So there's so many different steps. I mean, there's huge research needs. You know, I alluded to this tissue-specific methylation pattern. So I think there's a lot more that we need to learn about the cell types of origin of this cell-free DNA and what it can teach us about the underlying pathophysiologic process going on. And so, you know, my hope is that we can determine different rejection subtypes, different tissues of origin with cell-free DNA testing. And then, you know, I hope I showed that partnership with industry has been so important, because we can sit in our labs and do all the research we want and have really fascinating results, but it's never going to translate to patient care unless you partner with industry, right? And so that's something that I've kind of learned over the years is, you know, a lot of these companies have incredible research and development teams of really talented scientists who can help you then bring your assay that you've developed to clinical use. And they have the infrastructure to do that, getting FDA approval and the marketing and distribution and so on. So they're definitely very valuable partners. And then, you know, lately I've been learning more about health economics, which I don't necessarily want to, but CMS passed what's called a billing article at the end of 2023, basically saying that they'll no longer reimburse for doing more than one molecular assay at a time. And that caused huge upheaval in the transplant community, because if anything, we're going in the opposite direction where we want to do more molecular assays and get away from doing invasive procedures. And kind of, I learned about this a lot behind the scenes as I was involved in some advocacy work around this topic. But basically, they have a private contractor who they basically asked to kind of advise them on this decision. And the private contractor said, oh, we shouldn't pay for more than one test at a time. And that's the policy that CMS adopted, which is actually pretty short-sighted, and I think is going to really stifle further development and use of these non-invasive assays. So there's been a lot of, I went to Capitol Hill last December and, you know, spoke to Congress about kind of the importance of molecular testing and transplantation and how we should be able to do more than one test at a time. And so hopefully, you know, the policy will continue to evolve in that direction to fit this growing need.
Any other questions? I think we're getting right up to time. Thank you again for that
Thank you.
fantastic presentation.
Thanks so much, Tom.
The biopsy tree
Yes,
for
exactly.
the tumor.
One test fits all.
It is the dream.